Hello, chick, hello, hello.
Vande Mataram Shubhra Jyotsna Pulakitayaminim Fulla Kusumita Drumadala Shobhinim Suhasinim Sumadhura Bhashinim Sukhadam Varadam Mataram Vande Mataram Koti Koti Kantha Kala Kala Ninad Karale Koti Koti Bhuje Dhrita Kar Karwale Abla ke numaye tu bhale Bahubal Dharanim Namami Taranim Ripudal Varanim Mataram Vande Mataram Tumi Vidya Tumi Dharma Tumi Hriday Tumi Marma Tvam vi Pranah Sharire Bahute Tumi Ma Shakti Hridaye Tumi Ma Bhakti Tumare Pratima Gari Mandire Mandire Vande Mataram Tvam hi Durga Dasha Praharan Dharini Kamala Kamala Dal Viharini Vani Vidya Daini Namami Tvam Namami Kamalam Amalam Atulam Sujalam Sufalam Mataram Vande Mataram Shamalam Saralam Sushmitam Bhushitam Dharanim Bharanim Mataram Vande Mataram Vande Mataram
Thank you, Divya, Sanitya, and Anushri. Light symbolizes the knowledge, and the pursuit of knowledge is at core of science. Let us light the lamp of scientific temper, enshrine the spirit of rational thinking. May I request the dignitaries on the dais to kindly come forward and light the lamp. Thank you, sir. Thank you, ma'am. On February 28, 1928, Raman led an experiment with another Indian physicist, K.S. Krishnan, on the scattering of light. When he discovered the effect of the scattering of light when passing through different materials, what is now called as Raman effect. He discovered that an inelastic scattering of photons by matter, meaning that there is both an exchange of energy and a change in the light's direction. For this outstanding work, he was awarded the prestigious Nobel Prize in 1930. And in the year 1986, the Government of India adopted February 28 as a National Science Day to honor the scientific contributions of Sir C.V. Raman. Since then, National Science Day is celebrated in the nation to spread a message about the importance of science used in the daily life of a people, to display all the activities, efforts, and achievements in the field of science for human welfare. It is celebrated to discuss all the issues and to implement new technologies for the development of in the science field of science. Also, to give an opportunity to scientific-minded citizens in India. More than all, to encourage the people as well as popularize science and technology. The theme of this year's National Science Day is Global Science for Global Well-being. The Union Minister of State, Independent Charge, Science and Technology, Dr. Jitendra Singh, unveiled this theme on January 10, 2023, and said, as India enters 2023, the theme indicates India's emerging global role and rising visibility in the international arena. So now it is time to enter into the wonderland of proteins. May I request our director to kindly introduce the chief guest to the August gathering.
Elrigu Namaskara. A very good morning to all present here and greetings on the National Science Day. It is indeed a great pleasure for me to introduce today's chief guest, Professor Mattur Ramabhadras Shastri Narsimha Murthy, or who is fondly known as M.R.N. Murthy. He is a renowned crystallographer of India. Professor M.R.N. Murthy was born in the small village of Matturu, situated in Shimoga, Karnataka. And after completing his initial schooling at Shimoga, Professor Murthy joined the Central College at Begaluru as an honors student of physics. He completed his master's degree in physics from IIT Madras and did his PhD in crystallography under the guidance of Professor K. Venkatesan at the Indian Institute of Science. His doctoral work was partially concerned with conformation of peptides, where he determined peptide structures and carried out molecular mechanics calculation on model peptides to understand their conformational properties. These studies kindled his interest in proteins and macromolecular crystallography. Later, he did his postdoctoral work with one of the pioneers of structural biology, Professor M.G. Rossman of Purdue University, Indiana, USA, where he was involved in work on structural determination of glyceraldehyde 3-phosphate dehydrogenase and catalases. He has served as a faculty member at the Molecular Biophysics Unit at the Indian Institute of Science till his retirement. And during this period, he was one of the key persons in developing crystallography in India. For a very long time, he was the only crystallographer working on viral proteins in India. He has contributed about 100 protein crystal structures to the Protein Data Bank and has authored as many papers in internationally peer-reviewed journals. He has received several awards, and a few of them include honors such as being elected to the Indian Academy of Science in 1992, Shanti Swarup Bhattacharya Award, elected to the Indian National Science Academy 1996, Fellow of the National Academy of Sciences 2001, Rustam Choksi Award for Excellence in Science 2002, G.N. Ramachandran Commemoration Award 2003, Fellow of the Third World Academy of Sciences, Jagdish Chandra Bose Award for Life Sciences in 2005, AstraZeneca Distinguished Scientist Award for the Popularization of Science in 2005, G.N. Ramachandran Gold Medal for Excellence in Biological Sciences 2009, and at present, apart from many of the other awards that he has, at present he is Distinguished Professor and AstraZeneca Chair at the Institute of Bioinformatics and Applied Biotechnology, IBAB, in Begaluru. He is also Visiting Professor of IISr, Thiruvananthapuram. In addition to his passion for research, Professor Murthy has shown deep commitment to science education in the state of Karnataka. This interest has been facilitated by his intimate knowledge of the state language Kannada and his ability to express ideas of science with ease. He has actively participated in the activities of Karnataka Rajya Vigyana Parishad, an autonomous organization with the goal of promoting science education, Begaluru Association for Science Education, which is functioning from the Nehru Planetarium in Begaluru, Extension Lecture Program of the Indian Institute of Science supported by the University Grants Commission, New Delhi, and Karnataka State Council for Science and Technology. He has written several articles on scientific ideas and discoveries in the local language. Kannada Pustaka Pradhikara, a publishing body of the Karnataka government, has published a popular book on biotechnology written by him in Kannada. Indeed, it is a great fortune for us at CSIR-CFTRI to welcome him here today on this very special day for all scientists. And we do have a large number of students who would be gaining from the insights that Professor Murthy will be talking to us today. I once again welcome him. Thank you.
Thank you very much, Dr. Sridevi. I am very grateful to Sridevi, Purnima, Sri Rama for their wonderful hospitality. You know, this is a very special day for us. Science Day is more important than Navaratri or Christmas or Eid Milad. For us scientists, you know, this is the day we celebrate in the year. Our country will become a leader in the world only if all you young guys become wonderful scientists. First of all, many of you have the ability to become, Babas or Ramans or Ramanujans. You have the ability, but you have to work to develop it. If you do that, if every one of you try, some of you will succeed. There is no doubt about it. And then our country will be great. So we actually celebrate today as a festival. This is a festival of festivals for us science, for science, people working in science. I didn't know that a lot of young students are going to attend this talk. So I prepared the talk for audience which I thought would be CFTRI research scholars and faculty. So it is a little bit at a higher level, but I will try to reduce it and make it palatable to students as much as possible. ಅದು ಅಲ್ಲದೆ ಫಸ್ಟ್ ಆಫ್ ಆಲ್ ಇಲ್ಲಿ ಎಷ್ಟು ನಿಮಗೆ ಇಂಗ್ಲೀಷ್ ಬರುತ್ತೆ ಅಂದರೂ ಕನ್ನಡದಲ್ಲಿ ಹೇಳಿದರೆ ನಿಮಗೆ ಸ್ವಲ್ಪ ಜಾಸ್ತಿ ಬಂದಟ್ಟಾಗುತ್ತೆ. ಐ ಆಮ್ ನಾಟ್ ಗೋಯಿಂಗ್ ಟು ಸ್ಪೀಕ್ ಇನ್ ಪ್ಯೂರ್ ಕನ್ನಡ. ಅದು ಒಳ್ಳೆಯದಲ್ಲ, ಯಾಕೆಂದರೆ ಕೆಲವು ಕನ್ನಡ ಪದಗಳಿಗೆ ನೀವು ಇಂಗ್ಲೀಷ್ ಮೀಡಿಯಮಲ್ಲಿ ಓದ್ತಾ ಇದ್ದರೆ ಕೆಲವು ಕನ್ನಡ ಪದಗಳ ಪರಿಚಯವೇ ಇರೋದಿಲ್ಲ ನಿಮಗೆ. ಹೀಗಾಗಿ ಎರಡನ್ನು ಮಿಕ್ಸ್ ಮಾಡಿ ಮಾತಾಡ್ತೀನಿ. ಎಲ್ಲಾದರೂ ಗೊತ್ತಾಗದೆ ಹೋದರೆ ನೀವು ತುದಿ ಒಳಗೆ ಪ್ರಶ್ನೆಗಳನ್ನು ಕೇಳ್ಬೋದು, ಅದಕ್ಕೇನು ತೊಂದರೆ ಇಲ್ಲ. ಕಾಲರ್ ಮೈಕ್ ಇದೆಯಾ ನಿಮ್ಮ ಹತ್ರ? ಕಾಲರ್ ಮೈಕ್ ಹಾಕ್ಕೊಂಡು ಮಾತಾಡ್ತಿದ್ದೀನಿ. ಇದರಲ್ಲಿ ಮಾತಾಡಿದರೆ ನನಗೆ ಸ್ವಾತಂತ್ರ್ಯ ಇರೋದಿಲ್ಲ ಚಿತ್ರ ತೋರಿಸೋದಕ್ಕೆ. ನಾನು ತಿರುಗಿದರೆ ನನ್ನ ಧ್ವನಿ ಸಣ್ಣ ಆಗಿಬಿಡುತ್ತೆ. ಕಾಲರ್ ಮೈಕ್ ಹಾಕ್ಕೊಂಡ್ರು ಒಂದೇ ಸಮಯ ಇರುತ್ತೆ. ಆ ಚಿತ್ರದಲ್ಲಿ ತೋರಿಸಿದೆಯಲ್ಲ, ಅಲ್ಲಿ ತೋರಿಸಿರೋದು ಒಂದು ವೈರಾಣು, ಎ ವೈರಸ್ ಪಾರ್ಟಿಕಲ್. ಅಲ್ಲಿ ಹಲೋ, ಹಾಂ, ಈಗ ನಮ್ಮ ದೇಶದಲ್ಲಿ ಕಾಡಿಸ್ತಲ್ಲ ಸ್ವಲ್ಪ ಹಿಂದೆ ಕೋವಿಡ್ ವೈರಾಣು ಅಂತ. ದಟ್ ಆಲ್ಸೋ ಲುಕ್ಸ್ ಲೈಕ್ ದಿಸ್, ನಾಟ್ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಲೈಕ್ ದಿಸ್. This has a very regular shape, whereas COVID viranu doesn't have a regular shape. One looks like a round which is distorted here, salpa chapte irutte. It is not exact shape. How does it get its exact shape? I will ask you the question. How is it you have two eyes, not more? All of us have only two eyes. ಯಾರು ಶಿವಂತರ ಇಲ್ಲದ ಕಣ್ಣು ಇಟ್ಕೊಂಡು ಹುಟ್ಟೋಲ್ಲ. ವೈ? ದಿಸ್ ಈಸ್ ಅ ಕ್ವಶನ್ ವಿಚ್ ಹ್ಯಾವ್ ಟು ಆನ್ಸರ್. Science has answered this question. It is not that you have to find the answer. We know the answer. But we will see what we can do. What I want to talk to you today is the wonderful world of proteins. I am sure ನೀವೆಲ್ಲರೂ proteins ಅಂತ ಕೇಳೇ ಇರ್ತೀರಿ. Protein is very essential for our growth, right? If you don't have protein, you will get some kind of disease. But to do research on proteins is different. In fact, this institute is a food institute, which means ಯಾವ ಥರ ಆಹಾರ ತಿಂದರೆ ನೀವು ಆರೋಗ್ಯವಾಗಿರ್ತೀರಿ, ಯಾವ ಥರ ಆಹಾರವನ್ನೇ ನಾವು ರಕ್ಷಣೆ ಮಾಡ್ಕೋಬೋದು. This is the kind of research they do. You see, all these people throughout the year are working towards preservation of food, enhancement of food value. ಆ ಥರ we can do work on various aspects of protein. I will tell you some aspects, and I will also illustrate to you how exciting it is. See, science work ಮಾಡ್ತಾ, you don't know what you are going to get. See, it's a big secret. It is ಪತ್ತೇದಾರಿ ಕಾದಂಬರಿ ಥರ, ಯಾರು ಕೊಲೆಗಾರ ಅಂತ ಗೊತ್ತಿರಲ್ಲ. At the last, when you work for years, you will know the mystery. So how to unravel this mystery, that is the job of a scientist. And that is what I am going to illustrate to you by one or two examples. I have many examples here, but if I talk at a level that you understand, then I will only be able to cover one or two stories, okay? One or two mystery stories. We will see what mystery is there. So we want to understand something about proteins. Now, see, first of all, ಯಾರು ಪ್ರೋಟೀನ್ ಮೇಲೆ ವರ್ಕ್ ಮಾಡ್ತಾರೆ ಅಂತ ಈ ಇನ್ಸ್ಟಿಟ್ಯೂಟಲ್ಲಿ CFTRI ಅಲ್ಲಿ ಯಾತಕ್ಕೆ ಪ್ರೋಟೀನ್ ಮೇಲೆ ವರ್ಕ್ ಮಾಡ್ತಾರೆ? ಪ್ರೋಟೀನ್ಸ್ ಆರ್ ಬಯೋಮೆಟೀರಿಯಲ್ಸ್. ಸಿ, ಎವ್ರಿಥಿಂಗ್ ಇಸ್ ಎ ಬಯೋಮೆಟೀರಿಯಲ್. ವಾಟ್ ಎವರ್ ವಿ ಗೆಟ್ ಫ್ರಮ್ ಲೈಫ್ ಇಸ್ ಎ ಹುಲ್ಲು ಕೂಡ is a biomaterial. But proteins are very valuable biomaterials. How do you get protein? ನಮ್ಮ ದೇಹದಲ್ಲಿ ಪ್ರೋಟೀನ್ ಇದೆ, but it doesn't mean that you will get a protein. You have to somehow separate the protein. So, but once you separate, it becomes a biomaterial, and you can make many applications. Tumba useful applications na kanditkobodu. Then, of course, if you supply to the people, they will have use of this protein. Actually, you may have to study what kind of property the protein has. See, if I become your friend, I want to know what kind of a person you are. Same way, if you work with a protein, you want to find what kind of a protein it is. Is it positively charged, negatively charged in water or buffer? Protein can have positive or negative charge. It can be big, it can be small. What is the molecular weight of a protein? What does it interact? What does it do? What is the job of the protein? ಕೆಲವು ತುಂಟು ಹುಡುಗರಿರ್ತಾರೆ, ಬರೀ ಗಲಾಟೆ ಮಾಡೋದು ಅವ್ರ ಜಾಬು. What does this protein do? Does it cause a disease? Or ನಿಮಗೆ ಭೇದಿ ಉಂಟು ಮಾಡುತ್ತಾ? You know, actually there are proteins which cause diarrhea, toxins, you know, snake venom. ನಾಗರಹಾವು ಕಚ್ಚಿದರೆ ನಾವು ಸುತ್ತೋಗ್ಬಿಡ್ತೀವಿ. ಯಾಕೆಂದರೆ ಅದು ವಿಷ, ಅದು ಪ್ರೋಟೀನ್. ಅದನ್ನು ಸ್ಟಡಿ ಮಾಡ್ಬೋದು. ಸೊ ಯು ಸಿ, ಪ್ರೋಟೀನ್ ಹ್ಯಾಸ್ ಎ ವೆರಿ ವೈಡ್ ಅಪ್ಲಿಕೇಶನ್. ಅದನ್ನೆಲ್ಲ ಸ್ಟಡಿ ಮಾಡೋದು. ಹೇಗೆ ಯಾವ ಪ್ರೋಟೀನ್ ಇದ್ದ ನಾಗರಹಾವಲ್ಲಿ ನಾಗರಹಾವಿನ ವಿಷಯದಲ್ಲಿ ಯಾವ ಪ್ರೋಟೀನು ನಮ್ಮನ್ನು ಸಾಯಿಸುತ್ತೆ ಅಂತ ಅದು ಗೊತ್ತಾದರೆ ಅದಕ್ಕೆ ಆ್ಯಂಟಿಡೋಟ್ ಕಂಡಿಟ್ಕೋಬೋದು ಅಲ್ವಾ? ಅದು ದಟ್ ಈಸ್ ದ ಯೂಸ್. ಸೊ ಎವ್ರಿಬಡಿ ಈಸ್ ಇಂಟ್ರೆಸ್ಟೆಡ್ ಇನ್ ಪ್ರೋಟೀನ್. ಆ್ಯಂಡ್ ಜನರಲಿ ಪ್ರೋಟೀನ್ಸ್ ಆರ್ ನಾಟ್ ಟಾಕ್ಸಿಕ್. But of course, if you take snake venom, it is toxic. It's not all proteins are non-toxic, but most of the proteins are non-toxic, and they also can get degraded in our body. ಅದು ಹಾಗೆ ಇರೋಲ್ಲ. ನೀವು ಪ್ರೋಟೀನ್ ತಿಂದರೆ ಅಜೀರ್ಣ ಆಗೋಗ್ಬಿಡುತ್ತೆ. ಹಾಗೆ they don't cause inflammation. ಕೆರ್ತಾನೋ ಏನೋ ಮಾಡೋಲ್ಲ. So proteins are very good biosources which we can use for very many purposes. If you are clever, you can make money out of proteins. See, CFTRI has actually made, they don't want to make money, but they have developed products which you and me are enjoying today. ನೀವು ದೋಸೆ ಹಿಟ್ಟು ಕೊಟ್ಕೊಂಡಿದ್ದೀರಾ, ಇಡ್ಲಿ ಹಿಟ್ಟು, ನಂದಿನಿ ಇಡ್ಲಿ ಹಿಟ್ಟು, ರೆಡಿಮೇಡ್ ಇಡ್ಲಿ ಹಿಟ್ಟು, ಯಾರಾದರೂ ಗೊತ್ತಿಲ್ವ ಯಾರಿಗಾದರೂ? ನೆಕ್ಸ್ಟ್ ಟೈಮ್ ನಂದಿನಿ ಸ್ಟೋರ್ ಇರುತ್ತಲ್ಲ, ಅಲ್ಲಿ ಹೋದಾಗ ಕೇಳಿ ನೋಡಿ, ದೋಸೆ ರೆಡಿಮೇಡ್ ದೋಸೆ ಹಿಟ್ಟು ಇಡ್ಲಿ ಹಿಟ್ಟಿದೆಯಾ ಅಂತ. ಅದನ್ನು ಹೆಂಗೆ ಮಾಡೋದು, ಅದನ್ನು ಹೆಂಗೆ ಇಟ್ಕೊಳ್ಳೋದು? ಇವಾಗ ನಿಮಗೆ ಈ MTRದು ಹುಳಿಯನ್ನ ಸಿಗುತ್ತೆ ಒಂದು ಪಾಕೆಟಲ್ಲಿ. ಆ ಹುಳಿಯನ್ನೇನು ಒಂದು ತಿಂಗಳ ಮೇಲೆ ತಿನ್ಬೋದು ನೀವು. ಅದೇನು ಹಾಗೇ ಇರೋಲ್ಲ, ಹಾಳಾಗೇ ಇರೋಲ್ಲ. ಅದು ಹೇಗೆ? ದ್ಯಾಟ್ ಈಸ್ ಟೆಕ್ನಾಲಜಿ. ಯು ಸಿ ದಟ್ ಸಂಬಡಿ ಹ್ಯಾಸ್ ಡಿಸ್ಕವರ್ಡ್. ಆ ಥರ You see, if you can make these and make lot of advertisement, you can make lot of money. There are so many things in the market. And I am sure, I am almost sure, I cannot say, see, idu, hulugulu, insects. If you can eat insects, that's a very good food. Much better than eating, red meat. Red meat is tumba cholesterol irutte. Not good for health. Eating a lot of meat is not good for health. But, see, eating fish is okay. It's not bad, that bad. But eating insects is best. So you can make insect food. And maybe one of you will develop insect food and market it. You can actually sell it for a high price. Say it's beautiful food, it is a beautiful protein. Very rich protein, no cholesterol, no bad effects, no fat. Atara, you see, there are people experimenting on that. All this will come by research. There are poisonous insects. You cannot eat anything. See, mushrooms. Many people eat mushrooms. There are very poisonous mushrooms. So you have to know which mushroom is edible, which mushroom is not edible. That is scientific investigation. See, scientific investigation is you have to whole level marbekontilla. In everyday life, we encounter science. So that way, insect proteins are something that we can explore. We are a tropical country. We have a lot of insects. And if we can catch them or grow them in our home, then we can make insect food. That you can eat or that you can feed it to your cow or goat or sheep. We can also eat. It's okay. You see, food is just a matter of what we get used to. So if you start eating insects, it will be Yamamoto Indians eat insects all the time. And many countries in Africa actually eat food, not processed, just raw insects. They take a lot of insects and cook it and eat it. So it's not a bad thing. Don't feel very terrible about it. It's all what we are brought up with. That food seems to be all right for us. So you see, there is a lot of scope in all this research. And you can also make a lot of money, but you have to advertise a lot. You have to start a company. And sometimes you have to advertise and cheat people. There is no food value, but you say there is a lot of food value. I won't tell you about cheating, but there is cheating in the market. Anyway, see, actually you may not recognize, see, proteins can be used for several applications. Hesri heltini, someday you will realize what these are. See, this gelation, aggregation, micro and nanostructures, carrier molecules. Just I will tell you a little bit here, encapsulation properties. ಏನಪ್ಪ ಅಂದರೆ ಯಾವುದಾದ್ರೂ ಔಷಧಿ ತೊಗೊಂಡ್ವಿ ಇಟ್ಕೊಳ್ಳೋಣ. ಆ ಔಷಧಿ ತೊಗೊಂಡ್ರೆ ಅದು ಯಾವುದೋ ಒಂದು ಭಾಗಕ್ಕೆ ಹೋಗಿ ಕೆಲಸ ಮಾಡಬೇಕು. See, if your lungs are infected, you take a medicine, it should go to lungs and do its job. But actually it goes to all over the body. It goes from head to foot. And then it will have side effects because it is not going where the disease is. It is going everywhere. But nowadays you can develop carriers. ಏನು ಮಾಡೋದು ಅಂದರೆ ಒಂದು ಚೀಲ ಅದರೊಳಗೆ ಔಷಧಿ ಇಡೋದು. ಆ ಚೀಲ ಹೋಗಿ ಎಲ್ಲಿಗೆ ಹೋಗಬೇಕೋ ಅಲ್ಲಿಗೆ ಹೋಗುತ್ತೆ. ಇಟ್ ಗೋಸ್ ಟು ದ ಲಂಗ್ಸ್ ಆ್ಯಂಡ್ ದೆನ್ ರಿಲೀಸಸ್ ದ ಮೆಡಿಸಿನ್ ಸೊ ದಟ್ ಓನ್ಲಿ ಲಂಗ್ಸ್ ಗೆಟ್ ಟ್ರೀಟೆಡ್. ದ ಅದರ್ ಬಾಡಿ ಡಸಂಟ್ ಗೆಟ್ ಟ್ರೀಟೆಡ್. ಅವಾಗ ಟಾಕ್ಸಿಸಿಟಿ ಕಡಿಮೆ ಇರುತ್ತೆ. See, that's a very good thing, but it needs a lot of research. And proteins can be used as carriers of drug molecules. So obviously this institute will be interested in studying such proteins as carriers of medicinal compounds. I have shown here ಯಾವ ಪ್ರೋಟೀನು ಕಮರ್ಷಿಯಲಿ ವಯಬಲ್ ಅಂತ. ನೀವು ಯಾವ ಪ್ರೋಟೀನ್ ಮೇಲೆ ವರ್ಕ್ ಮಾಡಿದರೆ ದುಡ್ಡು ಮಾಡ್ಬೋದು ನೀವು. ಅಂದರೆ ನೀವು ಚೆನ್ನಾಗಿ ರಿಸರ್ಚ್ ಮಾಡಬೇಕು, ಆದಮೇಲೆ ದುಡ್ಡು ಬರುತ್ತೆ. ಇಲ್ಲಿ ನೋಡಿ, ಪ್ಲಾಂಟ್ ಪ್ರೋಟೀನ್ಸ್ ಇದೆ ಬೇಕಾದಷ್ಟು ಇಲ್ಲಿ. ಸಿ ಐ ವೋಂಟ್ ರೀಡ್ ಆಲ್ ಆಫ್ ದೀಸ್. ಪ್ಲಾಂಟ್ ಬೇಸ್ಡ್ ಪ್ರೋಟೀನ್ಸ್, ಇದೆಲ್ಲ ಪ್ಲಾಂಟಿಂದ ಬಂದಿರೋ ಪ್ರೋಟೀನ್ಗಳು. ಇದೆಲ್ಲ ಅನಿಮಲ್ಸಿಂದ ಬಂದಿರೋ ಪ್ರೋಟೀನ್ಗಳು. ಇದು ಎಲ್ಲದರಿಂದಲೂ ಯು ಕ್ಯಾನ್ ಆ್ಯಕ್ಚುಲಿ ಮೇಕ್ ಅಪ್ಲಿಕೇಶನ್ಸ್ ಆ್ಯಂಡ್ ಪ್ರೊವೈಡ್ ಯೂಸ್ಫುಲ್ ಥಿಂಗ್ಸ್ ಟು ಪೀಪಲ್. ನಿಮಗೆ ಗೊತ್ತು, ಎಷ್ಟೋ ನಾವು ಗಮನಿಸೋದಿಲ್ಲ. ಸಿಲ್ಕ್ ಬಟ್ಟೆ ಹಾಕ್ಕೊಂಡಿದ್ದೀರ ಯಾರಾದರೂ? ಸಿಲ್ಕ್ ಶರ್ಟು ಅಥವಾ ಲಂಗ. ಆ ಸಿಲ್ಕ್ ಎಲ್ಲಿಂದ ಬಂದಿದೆ? ಸಿಲ್ಕ್ ವರ್ಮ್ ಗೂಡು ಕಟ್ಬಿಟ್ಟು ಕೊಡುತ್ತೆ. ಆ ಸಿಲ್ಕ್ ವರ್ಮಿಗೆ ಕಾಯಿಲೆ ಬರುತ್ತೆ. ಅದು ಯಾವ ಕಾಯಿಲೆ ಬರುತ್ತೆ ಅಂತ ಕಂಡು ಹಿಡ್ಕೋಬೇಕು, ಅದಕ್ಕೆ ಕಾಯಿಲೆ ಬರದೇ ಇರೋ ಥರ ನೋಡ್ಕೋಬೇಕು. ಆಲ್ ದಟ್ ಈಸ್ ಸೈನ್ಸ್, ಯು ಸಿ. If you do that, there is a Sericulture Institute in Mysore. If you do that research, you can make. Do you know which country produces lot of silk? Huh? China. You see, our rival. If you want to beat China, you have to be very good silk scientist. One who works with silkworms and produces silk cheaper than what they do in China. That needs lot of scientific research. And maybe some of you will do. You see, this science has so much scope, you can almost do any subject and do very good work in science. And this will be a national service also. So silk is a big thing. So like that, there are many proteins from plants and animals which are commercially viable, which you can make profit out of them. Of course, you see, actually, now esto ಔಷಧಿ ತಗೋತೀವಲ್ಲ, sometimes we don't wonder how that medicine is working. ನೀವು ಜ್ವರ ಬಂದರೆ ಔಷಧಿ ತಗೊಳ್ತೀರಿ, ಟೆಂಪ್ರೇಚರ್ ರೆಡ್ಯೂಸ್ ಮಾಡೋದಕ್ಕೆ ಔಷಧಿ ತಗೋತೀರಿ, ತಲೆನೋವು ಬಂದರೆ ಔಷಧಿ ತಗೋತೀರಿ. ಹ್ಯಾಕೆ ಕೆಲಸ ಮಾಡುತ್ತೆ ಅದು? ಇಫ್ ಯು ವಾಂಟ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಹೌ ಮೆಡಿಸಿನ್ಸ್ ವರ್ಕ್, ಆ್ಯಕ್ಚುಲಿ ಯು ಹ್ಯಾವ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಪ್ರೋಟೀನ್ಸ್. Because what medicines or drugs do is to go to our body and bind to a protein. ಹೋಗಿ ಗಂಟು ಹಾಕೊಂಡ್ಬಿಡುತ್ತೆ, ಯಾವುದೋ ಪ್ರೋಟೀನಿಗೆ ಗಂಟು ಹಾಕೊಂಡ್ಬಿಡುತ್ತೆ. ಆ ಪ್ರೋಟೀನ್ ಏನೋ ಮಾಡ್ತಾ ಇರುತ್ತೆ. ಆ ಪ್ರೋಟೀನ್ ಮಾಡೋದನ್ನು ಜಾಸ್ತಿ ಮಾಡ್ಬೋದು, ಆ ಪ್ರೋಟೀನನ್ನು ಆ್ಯಕ್ಟಿವೇಟ್ ಮಾಡ್ಬೋದು, ಅಥವಾ ಆ ಪ್ರೋಟೀನನ್ನು ಡಿಆಕ್ಟಿವೇಟ್ ಮಾಡ್ಬೋದು. ಅಂದರೆ ನೀವು ಏನಪ್ಪ, ನಿಮ್ಮ ಫ್ರೆಂಡ್ ಇದ್ದಾನೆ, ನೀವು ಒಂದಷ್ಟು ಕಾಫಿ ಕುಡಿಸ್ಬಿಡಿ, ಅವ್ರಿಗೆ ನಿದ್ದೆನೇ ಬರೋಲ್ಲ, ಚೆನ್ನಾಗಿ ಕಾಫಿ ಕುಡಿಸ್ಬಿಡಿ, ಆ್ಯಕ್ಟಿವ್ ಆಗಿರ್ತಾರೆ. ಅದೇ ನೀವು ನಿದ್ದೆ ಗುಳಿಗೆ ಕೊಡಿ, ಮಲಗಿಬಿಡ್ತಾರೆ, ಆಮೇಲೆ ಏನು ಮಾಡೋದೇ ಇಲ್ಲ. ಆ ಥರ ವಿ ಕ್ಯಾನ್ ಆ್ಯಕ್ಟಿವೇಟ್ ಎ ಪ್ರೋಟೀನ್ ಆರ್ ಟರ್ನ್ ಆಫ್ ಎ ಪ್ರೋಟೀನ್, ಆ್ಯಂಡ್ ದ್ಯಾಟ್ ಈಸ್ ವೆರಿ ಯೂಸ್ಫುಲ್. ಬಿಕಾಸ್ ಆ್ಯಸ್ ಯು ಸಿ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಗಾಬಾ ಅಂತ ಒಂದು ಕಾಂಪೌಂಡ್ ಇದೆ. ನಮ್ಮ ಮೆದುಳಲ್ಲಿ ಗಾಬಾ ಇರುತ್ತೆ. ಆ ಗಾಬಾ ಇದ್ದರೆ ಹೆಲ್ತಿಯಾಗಿರ್ತೀವಿ. ಗಾಬಾ ತುಂಬ ಕಡಿಮೆ ಆಗಿಬಿಟ್ರೆ ಫಿಟ್ಸ್ ಬರುತ್ತೆ, ನಮಗೆ ಮೈಯೆಲ್ಲ ನಡುಗುತ್ತೆ, ಎಚ್ಚರ ಹೋಗುತ್ತೆ. ಅದು ಗಾಬಾ ಕಮ್ಮಿ ಆದರೆ ಆಗುತ್ತೆ. ಬೇಕಾದಷ್ಟು ರೀಸನ್ ಇದೆ ಫಿಟ್ಸ್ ಬರೋದಕ್ಕೆ, ಒಂದೇ ಕಾರಣ ಅಲ್ಲ. ಒಂದು ಕಾರಣ ಏನಪ್ಪ ಅಂದರೆ ಗಾಬಾ ಕಮ್ಮಿ ಆಗುತ್ತೆ. ಆದರೆ ಆ ಗಾಬಾ ತಯಾರಾಗುತ್ತಲ್ಲ, ನಮ್ಮ ದೇಹದಲ್ಲಿ ಯಾವುದು ತಯಾರಾದರೂ ಪರ್ಮನೆಂಟಾಗಿ ಇರಬಾರ್ದು, ಸ್ವಲ್ಪ ಹೊತ್ತು ಇರಬೇಕು. ಇವಾಗ ಹೊಟ್ಟೆ ಇದೆ ಇಟ್ಕೊಳ್ಳೋಣ, ಆಹಾರ ತಿಂತೀವಿ ನಾವು, ಅದನ್ನು ಜೀರ್ಣ ಮಾಡಬೇಕು. ಆ ಜೀರಕ ಎನ್ಸೈಮ್ಸ್, ಅವು ಪ್ರೋಟೀನು. The food will get digested because of proteins. They are called enzymes. Okay? Enzymes ನಮ್ಮ ಹೊಟ್ಟೆ ಒಳಗೆ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ. ನಾವು ಊಟ ಮಾಡದೇ ಇದ್ದಾಗ we don't want those enzymes because they may start chewing our own stomach. Because our stomach also is a biological material. It can get digested. So we want those enzymes when we eat food or just before we eat food. And then we don't want. ನಿಮ್ಗೆ ಗೊತ್ತಲ್ಲ, ಡಯಾಬಿಟಿಸ್ ಬಂದಾಗ ಅವರು ಊಟಕ್ಕೆ ಮುಂಚೆ ಒಂದು ಮಾತ್ರೆ ತಗೋತಾರೆ. ಆ ಮಾತ್ರೆ ತೊಗೊಂಡು ಊಟ ಮಾಡದೆ ಹೋದರೂ ತೊಂದರೆ, ಮಾತ್ರೆ ತೊಗೊಳ್ಳದೆ ಊಟ ಮಾಡಿದರೂ ತೊಂದರೆ. ಮಾತ್ರೆ ತೊಗೋಬೇಕು, ಊಟ ಮಾಡಬೇಕು. ನಮ್ಮ ದೇಹ ಅದನ್ನು ತಾನಾಗೇ ಮಾಡಿಬಿಡುತ್ತೆ. ಆ ಮಾತ್ರೆ ತೊಗೋತಾರಲ್ಲ ಅವರು, ನಮ್ಮ ದೇಹ ಏನು ಮಾಡುತ್ತೆ ಹೇಳಿ? ಊಟ ಮಾಡಿದ ಕೂಡಲೇ ಇನ್ಸುಲಿನ್ ಉತ್ಪತ್ತಿ ಮಾಡುತ್ತೆ. ಊಟ ಮಾಡದೆ ಹೋದರೆ ಇನ್ಸುಲಿನ್ ಉತ್ಪತ್ತಿ ಮಾಡೋಲ್ಲ. ಇನ್ಸುಲಿನ್ ಆಹಾರ ಸಕ್ಕರೆ ಕರಗಿ ಹೋಗೋದಕ್ಕೆ ಸಹಾಯ ಮಾಡುತ್ತೆ. ಸಿ, ಅವರ್ ಬಾಡಿ ಈಸ್ ಎ ವಂಡರ್ಫುಲ್ ಮೆಷೀನ್. ಆ್ಯಂಡ್ ಇಫ್ ಯು ವಾಂಟ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಹೌ ದಿಸ್ ಬಾಡಿ ವರ್ಕ್ಸ್, you have to know how proteins work. That is why you see it's a wonderland. Protein is a wonderland. Anyway, ಆ ಗಾಬಾ ಅಮಿನೋ ಟ್ರಾನ್ಸ್ಫರೇಸ್ ಅಂತ ಒಂದು ಎನ್ಸೈಮ್ ಇದೆ. ಆ ಎನ್ಸೈಮ್ ಗಾಬಾನ ನಾಶ ಮಾಡಿಬಿಡುತ್ತೆ. ಆಸ್ ಐ ಸೆಡ್, ಎವ್ರಿಥಿಂಗ್ ಹ್ಯಾಸ್ ಟು ಬಿ ಡಿಸ್ಟ್ರಾಯ್ಡ್ ಇನ್ ಅವರ್ ಬಾಡಿ. ಇಟ್ ಹ್ಯಾಸ್ ಟು ಬಿ ಮೇಡ್, ಇಟ್ ಹ್ಯಾಸ್ ಟು ಬಿ ಡಿಸ್ಟ್ರಾಯ್ಡ್. ಐ ವಿಲ್ ಟೆಲ್ ಯು ಅನದರ್ ಫ್ಯಾಂಟಾಸ್ಟಿಕ್ ಥಿಂಗ್. ಎನಿವೆ, ಆ ಗಾಬಾ ಅಮಿನೋ ಟ್ರಾನ್ಸ್ಫರೇಸ್ ಗಾಬಾ ನಾಶ ಮಾಡುತ್ತೆ. ನೋ ಯು ಸೆಂಡ್ ಎ ಮಾಲಿಕ್ಯೂಲ್ ವಿಚ್ ಇನ್ಆಕ್ಟಿವೇಟ್ಸ್ ಗಾಬಾ ಅಮಿನೋ ಟ್ರಾನ್ಸ್ಫರೇಸ್. ಅವಾಗ ಗಾಬಾ ನಾಶ ಆಗೋಲ್ಲ, ಆವಾಗ ಫಿಟ್ಸ್ ಬರೋದಿಲ್ಲ. ಸೊ ದಿಸ್ ಬಿಕಮ್ಸ್ ಎ ಮೆಡಿಸಿನ್ ಅಗೇನೆಸ್ಟ್ ಟು ಅವಾಯ್ಡ್ ಫಿಟ್ಸ್. ಸಿ ಹೌ ಸೈನ್ಸ್ ಕ್ಯಾನ್ ವರ್ಕ್. ಹಾಗೆ ಗೌಟ್ ಅಂತ ಒಂದು ಕಾಯಿಲೆ ಇದೆ, ಅದಕ್ಕೂ ಒಂದು ಡ್ರಗ್ ಇದೆ, ಅದು ಒಂದು ಪ್ರೋಟೀನ್ ಟ್ಯಾಬ್ಲೆಟ್ ಮೇಲೆ ವರ್ಕ್ ಮಾಡುತ್ತೆ. ಹಾಗೆ ಮೋಸ್ಟ್ ಆಫ್ ದ ಡ್ರಗ್ಸ್ ವಿ ಈಟ್ go and either activate a protein or turn off a protein, and that has a curative effect. How it works, what else does it do, all this has to be, you have to do research and understand, and that is what a protein scientist does. Well, actually these days there are many companies, you see, drug companies. how drugs are developed. Science is not static. This is what is wonderful for us. See, today if I do science, I started doing science 50 years ago. At that time, there were some problems in science which were very interesting. Many of them have been solved now. They are no more interesting. If I still keep doing what I was doing 50 years ago, people will call me a fool. I have to do new things. New techniques have come. So you have to adapt to new techniques. That way, it is not a static state, it is not like a pond. It is like a flowing river. It's always flowing, and you have to flow with the river. Atara, drug companies now have a strategy. That I won't go. It will take too much time to explain how drug companies work. There are many drug companies in Bangalore, and you can get a job in these drug companies. In fact, I work at an institute called IBAB. We take 40 students per year. Every one of them gets a job in a drug company, sometimes in India, sometimes abroad. So it is very important to get trained in protein science if you want to be in drug company. I won't, as I said, I won't go into that. And then I will tell you how important. I will want to start with CFTRI, this institute. How research problems will arise in the course of simple things. There is a pulse called Kesari dal. See, the name of the pulse is Kesari dal. Kelidira yaradru Kesari dal? Dal kelidira? Dal yatak ಮಾಡ್ತಾರೆ ಹೇಳಿ? ಚಪಾತಿ ಜೊತೆ ದಂಚ್ಕೊಂಡು ತಿಂತೀರಿ ಅಲ್ವಾ? ಚಪಾತಿ ಇದು ದಾಲ್ ಇಲ್ಲದೆ ಹೋದ್ರೆ ತಿನ್ನೋಕ್ಕಾಗತ್ತಾ? ಭಾಳ ಕಷ್ಟ. ದಾಲ್ ಇದ್ರೆ ಚಪಾತಿಗೆ ರುಚಿ, ಹೌದಾ? ಆ ದಾಲು ಬೇರೆ ಬೇರೆ ದಾಲ್ ಮಾಡ್ಬೋದು. ನೀವು ಹೆಸರು ಬೇಳೆ ಒಳಗೆ ಮಾಡ್ಬೋದು, ಕೇಸರಿ ದಾಲಲ್ಲಿ ಮಾಡ್ಬೋದು. ಇದು ಕೇಸರಿ ದಾಲ್, ಇಲ್ಲಿ ಕಾಣಿಸುತ್ತಲ್ಲ? ಈ ಕೇಸರಿ ದಾಲ್ ಏನಪ್ಪ ವಿಶೇಷ ಅಂದರೆ, ಮಳೆ ಅತೀ ಕಡಿಮೆ ಇದ್ದರೂ ಅದು ಬೆಳೆಯೋಕ್ಕಾಗತ್ತೆ. ನೀವು ಭತ್ತ ಬೆಳಿತೀರಲ್ಲ, ಭತ್ತ ನೀರಲ್ಲಿ ಕಟ್ಟಿರಬೇಕು. ಭತ್ತದ ಗದ್ದೆ ಒಳಗೆ ನೀರು ಇರಬೇಕು. ಅದ್ರ ಭತ್ತದ ಗದ್ದೆ ಯಾರಾದ್ರು ನೋಡಿದ್ದೀರಾ? ನೀರು ಕಟ್ಟಿರ್ತಾರೆ, ಅದರಲ್ಲಿ ನೀರಿರುತ್ತೆ. ನೀರಲ್ಲಿ ಚೆನ್ನಾಗಿ ಬೆಳೀತದು. ರಾಗಿಗೆ ನೀರು ಕಟ್ಟಿದರೆ ಕೊಳ್ತೋಗುತ್ತೆ. ರಾಗಿಗೆ ಎಷ್ಟು ನೀರು ಬೇಡ. ಈ ಸಿರಿಧಾನ್ಯ ಅಂತಾರಲ್ಲ, ಅದು ಯಾವುದಕ್ಕೂ ಜಾಸ್ತಿ ನೀರು ಬೇಡ. ಇದಕ್ಕೆ ತುಂಬ ಕಮ್ಮಿ ನೀರು ಸಾಕು. ಹೀಗಾಗಿ ತುಂಬ ಕ್ಷಾಮ ಅನ್ನುವಂಥ ಪರಿಸ್ಥಿತಿ ಒಳಗೂ ಕೇಸರಿ ದಾಲ್ ಬೆಳೆಯುತ್ತೆ. ಅದನ್ನು ಊಟ ಮಾಡಿದರೆ ನಮಗೆ ಹೊಟ್ಟೆ ತುಂಬುತ್ತೆ. ಹೀಗಾಗಿ ಬಡವರಿಗೆ ಕೇಸರಿ ದಾಲ್ ಭಾಳ ಇಂಪಾರ್ಟೆಂಟ್ ಆಗಿತ್ತು. ನಮ್ಮ ಇಂಡಿಯಾ, ಬಾಂಗ್ಲಾದೇಶ್, ನೇಪಾಳ್, ಇಥಿಯೋಪಿಯಾ, ಆಫ್ರಿಕಾದ ಕೆಲವು ದೇಶಗಳು ದೇವರ್ ಕನ್ಸ್ಯೂಮಿಂಗ್ ಕೇಸರಿ ದಾಲ್. ಬೆಳೆದು ಬಿಟ್ಟು ಕೇಸರಿ ದಾಲನ್ನು ತಿಂತಾಯಿದ್ರು. ವೆರಿ ಗುಡ್. ಇಲ್ಲಿ ನೋಡಿ, ಕೇಸರಿ ದಾಲ್ ಇಲ್ಲಿದೆ. ಅದರಿಂದ ಉಪ್ಪಿಟ್ಟು ಏನೋ ಒಂದು ಮಾಡಿದ್ದಾರೆ. ಅದು ನೋಡಿದರೆ ತಿನ್ನೋಣ ಅನಿಸುತ್ತೆ, ಅಷ್ಟು ಚೆನ್ನಾಗಿದೆ. ಆದರೆ ನೀವು ಕಾನ್ಸ್ಟೆಂಟಾಗಿ ತಿಂದರೆ ಹಾಗಾಗಿ ಹೋಗ್ತೀರಿ ನೀವು. ಕೇಸರಿ ದಾಲನ್ನು ತುಂಬ ಸಲ ತುಂಬ ದಿವಸ ತಿಂದರೆ ಈ ಥರ ಆಗ್ತೀರಿ. ಇದಕ್ಕೆ ಲ್ಯಾಥಿರಿಸಮ್ ಅಂತ ಕರೀತಾರೆ. ಇಲ್ಲಿ ನೋಡಿ ಹೇಗೆ, ಪಾಪ ಅವನಿಗೆ ಕಾಲೇ ಇಲ್ಲ, ನಡೆಯೋಕೆ ಆಗೋದಿಲ್ಲ, ಆ ಥರ ಆಗಿಬಿಡ್ತೀರಿ. ಯಾಕೆ ಹಾಗಾಗುತ್ತೆ ಅಂದರೆ, ಈ ಕೇಸರಿ ದಾಲಿಗೆ ಬಾಟನಿ ಒಳಗೆ ಒಂದು ಹೆಸರಿದೆ, ಲ್ಯಾಥಿರಸ್ ಸಟಿವುಸ್ ಅಂತಾರೆ. ಭಾರಿ ಭಯಂಕರ ಹೆಸರು. ಅದರ ಏನೂ ಇಲ್ಲ, ಕೇಸರಿ ದಾಲ್ ಅಂತ ಅರ್ಥ. ಅದು ಲಿನ್ನಿಯನ್ ನಾಮನ್ಕ್ಲೇಚರ್. ಲ್ಯಾಥಿರಸ್ ಸಟಿವುಸ್. ಅದರಲ್ಲಿ ಕೇಸರಿ ದಾಲ್ ಅಂತ ಹೇಳ್ತಾರಲ್ಲ, ಅದರಲ್ಲಿ ಒಂದು ಟಾಕ್ಸಿನ್ ಇದೆ, ಆಕ್ಸಾಲೈಲ್ ಅಮಿನೋ ಅಲನಿನ್ ಅಂತ ಒಂದು ಕಾಂಪೌಂಡು. ನಿಮಗೆ ಹೈಡ್ರೋಕ್ಲೋರಿಕ್ ಆ್ಯಸಿಡ್, ಸೋಡಿಯಂ ಕ್ಲೋರೈಡ್, ಅಮೋನಿಯಂ ಸಲ್ಫೇಟ್ ಅನ್ನೋ ಕಾಂಪೌಂಡ್ ಇರೋ ಥರ ಸ್ವಲ್ಪ ಮೋರ್ ಕಾಂಪ್ಲಿಕೇಟೆಡ್ ಕಾಂಪೌಂಡ್ ಇದು. ಒಂದು ಕೆಮಿಕಲ್ ಕಾಂಪೌಂಡ್. ಇಟ್ ಇಸ್ ಅ ಕೆಮಿಕಲ್ ಕಾಂಪೌಂಡ್. You can understand, water is a chemical compound. It is not an element. Water is H2O. Sugar is carbohydrate. That means carbon and C6O6H12. This is one sugar. Carbohydrate, carbon and water. ಆ ಥರ ಇದೊಂದು ಕಾಂಪೌಂಡು. ಬಟ್ ದಿಸ್ ಕಾಂಪೌಂಡ್ ಈಸ್ ಎ ಬ್ಯಾಡ್ ಗೈ. some compounds are good guys. Some compounds are very bad guys. This is a very bad guy. This compound is there in kesari dal. And if you keep eating kesari dal, that compound makes you lose control on your muscles all over the body. Not only kaalu, kai, ella hogibidutte. That is a terrible thing. So somebody had to find out. So actually here, you see, this is the I will show you this compound also. It's called beta-N oxalyl L-alpha beta diaminopropionic acid. Actually, name udda kanisute. See, diamino andre eradu NH3 group irutte anta amino group. Propionic acid, propane gottalla nimage. Methane, ethane, propane, butane. Adella hydrocarbons. Methane is CH4. C2H6, ethane. ಹಾಗೆ ಜಾಸ್ತಿ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. CnH2n plus 2 ಅಂತ ಫಾರ್ಮುಲಾ, ಜನರಲ್ ಫಾರ್ಮುಲಾ. You would have learnt it in your chemistry book. ಆ ಕಾಂಪೌಂಡ್ ಅಂತ ಸಿಂಪಲ್ ಕಾಂಪೌಂಡು, but it is a very dangerous compound. If you take that, you will have laterism. And Kesrizal has this compound. So if you can produce a Kesrizal without this compound, it will be a very good food. That was the idea with which research in this institute was started. Can we do something Kesrizal? First step, of course, is to detect what is the toxin and how is it synthesized. So actually, you see there are two very eminent people here. This is Rao, Professor Rao, who was a director of this institute. And this is Vijayalakshmi, who became a very famous neurologist of India, one of the greatest neurologists of India. She was a PhD student in this institute. They started this project, and I have shown here, see, many of the papers they published. It's not my paper. There, Rajgopal Rao and Vijayalakshmi's papers. You see, I think you won't understand the significance of this. Whenever you do a scientific research, eventually you have to publish it. If you don't publish it, it has no value. If you say, I have discovered a proof of Fermat's last theorem, or whatever it is, very difficult problem, Fermat's last theorem is a very difficult problem. You say, I have solved it. Nobody believes. You have to publish it. When you publish it, other people will read your paper and see whether your paper makes sense. Only when it makes sense, they publish. So actually, if they have published in a good journal, it means they have done a valid research. So that way, all this is valid research. So they have published many papers on this toxin. Of course, you see, it takes a long time to produce a crop. This research doesn't produce a crop without the toxin. That needs a lot more work. But this is a beginning. So nobody has come to the end. You have to do more. And then what happened, they're all our friends, you see, Vijayalakshmi, Rajgopal Rao, everybody were our friends. So actually, there were some studies on this toxin, not only by these people. See, this toxin is also found in silkworm. See, the same toxin that is in a dal is also in silkworm. See, you may wonder how it is possible. See, whatever proteins are there in our body, the equivalent proteins are in the pig that roams in the street. Do you know? In a way, pig and we are very related. We have descended from the same ancestor. We are evolutionarily related. But anyway, this protein is found in various organisms, and various people around the world have worked on this compound. And then, you see, actually, I have shown you the formula of this compound. That is the compound. You see, that this is not the compound. It's a small variation of this will produce the toxin. This is diaminopropionate. Oxalyl group is not there. It is diaminopropionate. See, propionate is a 3-carbon compound. Propionate andre carbon, carbon, carbon. The first carbon ige 3 hydrogen irutte. Second carbon ige 3 hydrogen irutte. Madhyad carbon ige 2 hydrogen irutte. Yakendre every carbon has to have 4 single bonds. You know, carbon has valency of 4. That you have learned in your school. So it's propionate formula CH3, CH2, CH3. It's a very simple compound. Adakke ee amino group sersidre illi it becomes diaminopropionate. There is an enzyme which was actually isolated in CFTRI. That enzyme, if you add it to this compound, it will reduce it to this compound. How does it, you see, this protein is involved in the synthesis of the toxin. ಆ ಟಾಕ್ಸಿನ್ ಸಿಂಥಸಿಸ್ ಮಾಡೋದಕ್ಕೆ ಈ ಪ್ರೋಟೀನ್ ಬೇಕಾಗುತ್ತೆ. What does this protein do? How does it do it? What is its structure? Is a question we can investigate. As I said, this is also part of the investigation towards producing a crop which has no toxin. But this is not the end. This is only a middle step. So just by doing this research, you won't have a plant. And I am not a plant biochemist, so I cannot even produce. But I can produce some data which can be useful at some point of time. So we decided, my subject, I will tell you in a minute what my subject is. We can do its structure. But you see, really, because you see, you young people have come, this would have been trivial for CFTRI. I just have to flash this slide, they know what it is. But unfortunately, my nature is such that I cannot just rush if I think that you don't know what I am talking about. So I will have to slow down. But I will cover much less, maybe only one story. But it's worth covering one story where you understand, young people understand, rather than covering 4 stories where I just talk over the head of all of you. Really, see, proteins are workhorses. See, what are proteins? They are workhorses. They do all the work in our body. See, there are different kinds of molecules in our body. You have heard of nucleic acids? Nucleic acids, what do they do? What do nucleic acid do? Any student can answer. What do nucleic acid do? Nucleic acid, you have heard of nucleic acid. DNA is a nucleic acid. What does it do? Yes, louder. Huh? It consists of genes. But what do genes do?
It transfers the inheritance of
It actually codes for proteins and many other molecules. But it also tells you how big my nose should be. See, everything we are is because of our DNA. All our genetic properties are decided by DNA. If the DNA is defective, you may have a genetic disorder. DNA is the master template of what we are going to be. So actually, if you take coronavirus, it's not a DNA there, it's a nucleic acid, but it's called RNA. That RNA is going to tell how devastating it is. If that RNA changes, coronavirus changes. Actually, we had original Chinese variant of the coronavirus. That was dangerous, but not that dangerous. Then something changed in its gene, in the nucleic acid. Then you had a Delta variant, which was extremely virulent. It killed actually tens of thousands of people in India. Then we had Omicron. That is another variant of the same virus. That was not as killer virus. It only made people ill for months, but it won't kill as much as Delta variant. So nucleic acid decides what kind of life we are going to lead, what kind of life a virus is going to lead, what kind of life a pig is going to lead. All of it is decided by DNA. But DNA is like your Sridevi, director. She will direct what the institute should do. But the actual work is done by scientists in CFTRI. And those scientists are the proteins of the cell, which means DNA is the director of the cell, proteins are the workhorses of the cell. Understand the difference. Proteins do all the work. What does this protein do? Sometimes proteins don't function by themselves. ಇವಾಗ ಕುದುರೆ ರೈಡ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಏನು ಮಾಡಬೇಕು? ಕುದುರೆ ಒಳಗೆ ಹತ್ತಿಬಿಟ್ಟು ರೈಡ್ ಮಾಡೋಕ್ಕಾಗತ್ತಾ? ಕುದುರೆ ಮೇಲೆ ಹತ್ತಿಬಿಟ್ಟು ಏನು ಮಾಡಬೇಕು? ಕುದುರೆ ರೈಡ್ ಮಾಡೋದು ನೋಡಿದ್ದೀರಾ? ರೇಸಲ್ಲಿ ಕುದುರೆ ರೈಡ್ ಮಾಡ್ತಾರಲ್ಲ, ಸುಮ್ಮನೆ ಕುದುರೆ ಮೇಲೆ ಕೂತು ಓಡಿಸ್ತಾರ? ಏನು ಮಾಡಿರ್ತಾರೆ? ಅದಕ್ಕೆ ಹಗ್ಗ ಕಟ್ಟಿರ್ತಾರೆ ಮೂಗಿಗೆ, ಅದನ್ನು ಹಿಡ್ಕೊಂಡಿರ್ಬೇಕು. ಅದಿಲ್ಲದೆ ಓಡಿಸೋಕ್ಕಾಗಲ್ಲ ನಿಮಗೆ. ಹಾಗೆ ಕೆಲವು ಪ್ರೋಟೀನ್ಗಳಿಗೆ ಏನೋ ಒಂದನ್ನು ಸೇರಿಸಬೇಕು, ಇನ್ನೊಂದು ಮಾಲಿಕ್ಯೂಲನ್ನು. ಇಲ್ಲದೆ ಹೋದರೂ ಪ್ರೋಟೀನ್ ಕೆಲಸ ಮಾಡೋಲ್ಲ. ತುಂಬ ಪ್ರೋಟೀನ್ಗಳಿಗೆ ಒಂದು ಮಾಲಿಕ್ಯೂಲ್ ಸೇರಿಸ್ಬೇಕು, ಈ ಸ್ಮಾಲ್ ಮಾಲಿಕ್ಯೂಲ್. ಇದಕ್ಕೆ ನಾವು ಪಿರಿಡಾಕ್ಸಾಲ್ ಫಾಸ್ಫೇಟ್ ಅಂತ ಕರಿತೀವಿ. ಪಿರಿಡಾಕ್ಸಾಲ್ ಫಾಸ್ಫೇಟ್ ಈ ಮಾಲಿಕ್ಯೂಲ್ ಸೇರಿಸಿದರೆ ಆ ಪ್ರೋಟೀನ್ ಆ್ಯಕ್ಟಿವ್ ಆಗುತ್ತೆ, ಅದ್ರ ಕೆಲಸ ಮಾಡುತ್ತೆ. ಆ ಪಿರಿಡಾಕ್ಸಾಲ್ ಫಾಸ್ಫೇಟನ್ನೇ ಉಪಯೋಗಿಸ್ಕೊಂಡು ಫಂಕ್ಷನ್ ಮಾಡುವಂಥ ತುಂಬ ಪ್ರೋಟೀನ್ಗಳಿದೆ. ಹೀಗಾಗಿ ಆ ಪ್ರೋಟೀನ್ಗಳೆಲ್ಲ ಹ್ಯಾಕೆ ಕೆಲಸ ಮಾಡುತ್ತೆ ಅನ್ನೋದು ಒಂದು ಜನರಲ್ ಕ್ವಶನ್ ಆಗುತ್ತೆ. ಅದಕ್ಕೆ ನಾನು ಆ ಪಿರಿಡಾಕ್ಸಾಲ್ ಫಾಸ್ಫೇಟು ಎಷ್ಟು ವರ್ಸಟೈಲ್ ಅಂತ ಇವಾಗ I will tell you, because you see, I didn't expect that you young people would be here in the audience. Now, rakta, yeah, what is the purpose of having red blood cells? What do red blood cells do? Red blood cells are made up of mainly a protein, and that protein is called hemoglobin. ನೀವು ಬ್ಲಡ್ ಟೆಸ್ಟ್ ಮಾಡಿಸಿದ್ರೆ ಹಿಮೋಗ್ಲಾಬಿನ್ ಪರ್ಸೆಂಟೇಜ್ ಬರೀತಾರೆ. ಎಷ್ಟು ಪರ್ಸೆಂಟ್ ಇರಬೇಕು ನಿಮಗೆ ಅದನ್ನು ತಿಳ್ಕೊಳ್ಳಿ. ಅದನ್ನು ನೀವು ಚರ್ಚೆ ಮಾಡೋದು ಬೇಡ. ಹಿಮೋಗ್ಲಾಬಿನ್ ಏನು ಮಾಡುತ್ತೆ? ವೈ ಹಿಮೋಗ್ಲಾಬಿನ್ is necessary? Yes, transport oxygen. But hemoglobin protein is incapable of transport oxygen unless a small molecule called heme is attached to it. That heme helps hemoglobin to carry oxygen. ಹಾಗೆ ಕೆಲವು ಪ್ರೋಟೀನ್ಗಳು ವರ್ಕ್ ಮಾಡಬೇಕಾದ್ರೆ ಈ ಪಿರಿಡಾಕ್ಸಾಲ್ ಫಾಸ್ಫೇಟ್ ಅದಕ್ಕೆ ಅಂಟ್ಕೊಂಡಿರಬೇಕು. ಆ ಪಿರಿಡಾಕ್ಸಾಲ್ ಫಾಸ್ಫೇಟ್ ಅಂಟ್ಕೊಂಡ್ರೆ those proteins can be made to do many different functions. See, hemoglobin will only carry oxygen, nothing else. Whereas PLP enzymes, PLP-dependent enzymes, they can do many things. ಬೇಕಾದ್ರೆ ಮನೆ ಕಟ್ಟುತ್ತೆ, ಇಲ್ಲೋದ್ರೆ ಮನೆ ಬಿಡಿಸುತ್ತೆ, ಇಲ್ಲೋದ್ರೆ ಸೋಫಾ ಅರೇಂಜ್ ಮಾಡುತ್ತೆ ಮನೆಯಲ್ಲಿ, ಆ ಥರ ಡಿಫ್ರೆಂಟ್ ಫಂಕ್ಷನ್ಸ್. So this protein does different biological functions. PLP enzymes. Whether PLP attached ಆಗಿರೋ enzymes, they will do all kinds of jobs in our body. So they are very important class. So we wanted to determine the structure. See, I have actually, there are lots of interesting things here. It is so interesting. I will spend all my, I don't know, Purnima has to tell me when I have to finish. Because you see, unless I am alerted, it's not going to finish. There is so much interest in every one of these slides. I don't want to skip and lose the interest. See, you know evolution, you have heard of evolution. What is evolution? Is it Manganinda Manava? That's absolute nonsense. No, that's not evolution. What is true? Have you gone to Mysore Zoo? Have you seen a chimpanzee there? You know that chimpanzees, Amma, your Amma, their Amma, their Amma, their Amma, their Amma. ಹೀಗೆ ಹೋಗಿ 50,000 ಜನರೇಷನ್ ಹೋಗಿ ಒಬ್ಬ ಅಮ್ಮ ಇದ್ರು ಅಲ್ಲಿ, ಗ್ರ್ಯಾಂಡ್ ಅಮ್ಮ, 1 millionth ಅಮ್ಮ. Actually, the chimpanzee is descendant from that Amma. I am also descendant from the same Amma. So we are related. You see, the chimpanzee is my cousin or uncle or nephew or niece. Some relation is there, but it is 1 millionth relation. See, ಚಿಕ್ಕಪ್ಪ ದೊಡ್ಡಪ್ಪ ಮಕ್ಕಳು ಫಸ್ಟ್ ಕಸಿನ್ಸ್. ಸೆಕೆಂಡ್ ಕಸಿನ್ಸ್ ಗೊತ್ತಾ ನಿಮಗೆ? ಫಸ್ಟ್ ಕಸಿನ್ ಅಲ್ಲ, ಸೆಕೆಂಡ್ ಕಸಿನ್ನು. ಸೆಕೆಂಡ್ ಕಸಿನ್ ಅಂದ್ರೆ ಗೊತ್ತಲ್ಲ, ಸ್ವಲ್ಪ ದೂರ ಆಗುತ್ತೆ ಸಂಬಂಧ. ಅಂದರೆ ಈಗ ಅಜ್ಜ ಅಜ್ಜಿ ಒಂದೇ ಇದ್ದರೆ ಕಸಿನ್ಸ್ ಆಗ್ತೀವಿ ನಾವು. ಮುತ್ತಜ್ಜಿ ಒಬ್ಬರೇ ಇದ್ದರೆ ಸೆಕೆಂಡ್ ಕಸಿನ್ಸ್ ಆಗ್ತೀವಿ. ಥರ್ಡ್ ಕಸಿನ್, ಫೋರ್ತ್ ಕಸಿನ್, maybe millionth cousin. Actually, this pig on the street also is our ancestor. We have common ancestry, right? See, that is what Darwin's theory is all about. Every organism in this world has come from the same ancestor. We all have come from Africa, no matter where you live in the world. Our original home is Africa. We are all immigrants, you know. Now we may be Indians, but at some point of time, our ancestors have walked all the way from Africa to India. But anyway, that's what Darwin's theory tells. But you see, it is also now possible, because of developments in modern biochemistry, we can ask how proteins have come. How does hemoglobin come into life? Where is the origin of hemoglobin? See, this is not evolution of animals. It is evolution of proteins. That we can investigate. This slide shows something about evolution of PLP-dependent enzymes. You see, you can learn very interesting mysteries here. Which protein has evolved into which other proteins? How did it evolve? At what point of time, how many million or billion years ago this event occurred? All this can be worked out using modern biochemical tools. You know, usually research in evolution was based on fossil finding. See, if you go to Hyderabad planetarium, there is a full fossil of a dinosaur. Once upon a time, dinosaurs roamed the Indian land, and they found a whole dinosaur bone, fossil bone, and they have brought to the planetarium and assembled it into a very nice skeleton. That you can see, if you go to Hyderabad, you can see. Evolution has created many organisms, exterminated many organisms. So it has created humans. Will it also exterminate humans? It looks very likely if you look at the war scenario in the world. But if you want to save this world, you have to learn science. You have to know what kind of action is going to kill all of us. It's a very, very great possibility. You know, in 2 generations, everybody may start dying in large numbers. Whole world may not die, but huge number, millions may die in all countries if we are not careful. You know, we have so much power. So we have to learn science. But anyway, this tells you something about how these enzymes themselves are evolved. Then, you see, I am going to skip this slide. So we came to CFTRI about 20 years ago. We took this protein, which actually synthesizes the toxin in kesari dal. See, kesari dal has a toxin, and that toxin is synthesized by an enzyme. And we took this enzyme. The name of the enzyme is diaminopropionate ammonia lyase. Doesn't matter, it's a protein. See, if you don't remember the name, it doesn't matter. It's a protein. What does this protein do? It will produce as a toxin in the dal. And so, if you study this protein, maybe you can see how you can avoid the toxin production in the dal. Just this study will not allow, but you have to make more studies. But it is one step in that direction. So we took this crystal, we took this protein. See, my own field, I am now coming to my field. My specialization is called crystal structure determination. What it means is that you take any material, this could be a protein, you purify a protein. See, this is an art, you know, it's absolute. Anything you study in science is absolutely fascinating. You know, if you take a little bit of, you know, cut here and take a little bit of my meat, how many proteins it has? How many proteins might be there in a little bit of a tissue? Plant or animal? You can take a leaf. You take a leaf and see how many proteins are there. There will be probably tens of thousands of proteins. But you have to purify one protein. You know, sometimes you see rice has some small stones. You separate the stones because you don't want to eat rice, you know, anna, with small stones. It hurts your teeth. You have to remove. How do you remove? Do you know how stones are removed from rice? Well, if you don't know, you go and ask your parents how you can remove small stones from rice. There is a method. It's a scientific method. But that method is going to separate stone and rice. Only 2 things. But here we have 10,000 proteins and we have to separate one protein. It is not a simple thing. So we have lots of experimental procedures developed over the last 70 years. Before that, people did not know how to purify a protein from mixture of 1,000 proteins. But today, you can purify anything that you want. You have so-called chromatographic procedures. So we did that and purified that protein. And then we made crystals of that protein. You know, just like sodium chloride or ammonium sulfate crystals, we grew crystals of a protein. And then we took that crystal to an X-ray lab, put the crystal and shoot it with X-rays. When you shoot a crystal with X-rays, it will scatter X-rays in certain directions. Not all directions, only certain specific directions. You can see those directions here. See, in different directions, reflections have come and made an imprint on the film. If you now measure the intensity of all these spots, tens of thousands of spots, you have to measure the intensity. And using that data, you will be able to eventually reconstruct how the protein looks. What is the structure of the protein? What is the atomic structure? Do you know water structure? You have an oxygen atom and 2 hydrogen atoms here. It's a bent molecule. Water is not oxygen, 2 hydrogens. It's not linear. Oxygen, 2 hydrogens. This angle is 105 degrees. Oxygen, water is a bent molecule. Like that, what is the molecular structure of the protein which has 5,000 atoms? I know exactly how they are connected. What is the structure of the protein? That is determined using these intensities. But that is a problem. See, this shows how biochemistry, how powerful biochemistry is. See, this is, you won't understand this, but very soon, within 2 or 3 years, you will understand this. This is what we call an electrophoresis experiment. And the peak here you see is a single protein. And that is a protein which produces the toxin, related to the toxin. And that protein can actually be purified in a very pure form. And you can make crystals. This is an experiment to show how pure our protein is. But let's not worry about that. And then this guy, Rajaram, made crystals of that protein. He made crystals of that protein. And he collected X-ray diffraction data. But you see, just X-ray diffraction data will not be enough to solve the structure. You need something more. There's something more. See, this is an X-ray diffraction pattern. How beautiful it is. See, as I said, any branch of science, anything you do is just thrilling. Whenever I go to the cold room and develop the photograph, dark room and develop the photograph, bring it and see, I cannot help but sing, twinkle, twinkle, little star, how I wonder what you are. All these star-like things are reflections from the crystal. And you have to measure the intensity of each one of them. See, not for this structure, for some other structure, we have measured 5 million reflections. So, you see, it's a big task, but you can measure. Well, just that is not enough. See, these reflections are X-rays. X-rays are waves. You know wave. You understand wave, and waves are like this. Waves have an amplitude. You know, tsunami wave you have found. That's a very big amplitude that can actually topple buildings. So it has an amplitude. That is related to intensity, how intense that reflection is. But that also has so-called phase. That is relative to one reflection. If one reflection has its high point here, crest, other reflection, is the crest here or slightly forward or backward? That is what we call phase. Whether the two reflections are in phase. If you are walking with your friend, when you put your right hand forward, will your friend also put right hand forward? Then we say you are in phase. But when you put your right hand forward, your friend is putting left hand forward, then you are actually out of phase. You can be in any different phase. And that phase also is necessary to solve the structure. But to do that, what we have to do is, I will give you a simple example. There is a symphony going here. All of these people are playing music. You are a blind person. I stand here, blind person, and I listen to this music. You see, I see the sound of, see, if I stand here, I will see these violins, very high sound. Whereas those viola, I will see less sound. But if I come here, I will hear drums more. Just by hearing all of this, will you be able to figure out who are the people playing the music? That is the question. But that you cannot answer unless you send a secret agent here. And that secret agent also plays music. Now again I stand at everywhere and listen to the music, how loud it is. But now I have the effect of all these players plus my secret agent. If I do that, then I will be able to figure out the phases. In other words, you have to have reflections from your protein crystal and reflections from protein crystal into which you have added a heavy metal atom. This is what is necessary. But unfortunately what happened is that whenever Rajaram prepared, see, this is native protein, which means no heavy atom. And then he put a cesium atom, and then bromine atom. Like that, he added heavy atoms. then you see the position of those musicians should not change. You send your secret agent, the secret agent should not disturb the position of the other players. But actually, you see, I won't tell you what this is. These numbers tell you that when he introduced heavy atom, the position of the musicians got changed, which means these are not useful for structure determination. So he couldn't solve the structure. 10 years we waited because there had to be developments in another field of science. And you see, actually long, long ago, two people, Ramasheshan, who became a director of Indian Institute of Science, and Professor Venkateshan, when these people had worked in 1950s and shown that if you can collect X-ray diffraction data at two different wavelengths, you know, X-rays have a wavelength, if two different wavelengths, you know, radio stations have a wavelength, you can tune it with your tuner, whichever station you want. Like that, these people showed that if you can collect X-ray diffraction data at two wavelengths, that will allow you to solve the structure in some complex way. But all X-ray machines in the laboratory produce only one wavelength. So there was no way we could collect. Then, you see, physicists developed a machine. You see how science is related? One subject gets related to another. Physicists developed this machine. This is called a synchrotron. This machine is so big that if you want to take a picture of the machine, you have to go in a helicopter and take a picture. This is more than a kilometer, this ring. And in this ring, there's a tunnel. The whole tunnel is highly evacuated. And electrons go round and round this tunnel with tremendous speed. When they go, they produce X-rays of all wavelengths. So only such a machine will produce all wavelengths. Our machine in our lab will not produce. This machine was built in a city called Grenoble in France. Such machines were built not in India, but in Japan, France, United States, some countries, such machines were built. We took, you know, we bought some machine time in France, and we could send the crystals, and we could collect the data. And then after 10 years, we could actually solve the structure. So I have some details of how we collected the data, don't worry. And these two ladies were involved in the structure solution. You see, this is the structure of the protein. How beautiful this structure is. But it is a simplified diagram. See, if you rake water, you have oxygen and two hydrogen. Like that, if I do the atomic structure of the protein, there will be 5,000 atoms, and it will be a jungle of atoms. We don't make head and tail. But we know that proteins are made up of polypeptide chains. You know, it's a linear chain. Even DNA is a linear chain. But DNA chain goes round and round. It is double helical. You would have, some of you at least would have seen double helical structure of DNA. But proteins wind in a very complex way. And it only shows how the protein is winded. It won't show the full structure. This is what we call a cartoon diagram of the protein. And there you have pyridoxal phosphate. That is the one that activates this protein, exactly like heme group activates hemoglobin. And you know, this protein, it tells you so much, this protein structure tells you so much. It's a very rich source of information. See, there is one color domain. This is a domain. Here, polypeptide is wound like this, like a spool. Then it goes elsewhere and winds again. So you have two domain structure for this protein, and in between the domain, pyridoxal phosphate binds. And you see, I won't tell you much about this. You see, this is how you see the structure of the protein. It's beautiful. You know, you look at the, once you solve the structure, you look at the structure. You know, you see the polypeptide going everywhere. You see every atom of your protein. It's an absolutely thrilling experience. You know, the day you solve the structure, you have in all grand 5,000 atoms sitting before you, making a big protein structure. Then, of course, you have to find out how it works. See, you have found a car. Car is beautiful. It has wheel, it has engine, everything. You have seen all of it. But how does it work? That is not simple. Well, of course, these students did a lot of experiments. I will skip this. It will be too much for you. But anyway, you see now, they can suggest exactly how see this molecule here, there is an amino acid coming here, and how that amino acid is chemically transformed. We call it enzyme mechanism. See, I will not see, this is for you young students, enzyme mechanism is a term, let it be in your head. It just tells you how exactly it works. See, your mother tells you, go and buy a dozen banana, and then go and buy some carrot, and then go and buy some, safety pins. Like that, she will give you a list. How do you do that list? Which shop will you go first? What is the sequence in which you do? Any chemical transformation is like that. See, hydrogen peroxide, H2O2, hydrogen peroxide. It will kill bacteria. So it is used to wash wounds. If you get a scratch, you wash it with hydrogen peroxide, H2O2. But that can be converted to H2O. Oxygen goes away, when the oxygen goes away, it becomes water. How does it do that? How does hydrogen peroxide become water? It is a rearrangement of atoms. How does rearrangement take place? That is what we call mechanism of action. So how does this enzyme work? All that mechanism has been worked out. Well, I think I am almost to the end of my time. See, I tell you, I mentioned I will tell you 4 stories. But I would have told 4 stories if the audience were purely from CFTRI. But now that you have young people are here, I cannot tell you 4 stories. I have come to the end of my time. See, 1 story I could tell, but it's okay. But I want to tell you, this is my concluding remarks. See, I want to tell you how unexpectedly you come to new streets. You see, all of a sudden you are walking in a mountainous region. All of a sudden you come to a spot where you see a deep gorge and beautiful mountain range opposite to you. That experience of beauty is something that happens all the time in science. That is what I want to impress on you. See, I actually want another story I wanted to tell you is a story which unfolded by itself. See, I have a friend in Holland called Wim Hof, very talented person. He was working on an enzyme called serine hydroxymethyltransferase, which is actually an enzyme. If you inhibit this enzyme, it will cure cancer. So it's a cancer target. And Appaji Rao in Indian Institute of Science was also working on that. Since those two were my friends, we started working on SHMT. And, actually, it has a very fantastic story. This guy is the one who solved the structure of serine hydroxymethyltransferase. He was my student, now he is my boss. He is the director of IBAB. I work in IBAB as an ordinary person, so he is my boss. See, it's very nice. A student becomes your boss. Okay, and then, you see, that experience was wonderful. Like that, I have other experience. These 2 guys, I have traveled all around India hunting for viruses, which are very suitable for diffraction studies. I have gone to every institute, agricultural college in India, trying to see whether they have viruses. Finally, we found one virus in Tirupati, and that is the virus structure. And there is a whole story behind it, but unfortunately, we cannot do all this. And this is also a fantastic story. It's a detective story, which this fellow did. I skipped that. And this is another friend of mine from Finland. With him, we have done some very nice work. I cannot tell you this. But in summary, this is Science Day. So actually, this is a day we celebrate. This is more important for us than any other festival of the year. And on this day, you determine that you will be an excellent scientist. If you determine and study for it, you will be an excellent scientist. So that's how I would like to end, wishing all the young students here all the best in their education, in their research career. And I want to express at the end, have confidence. That is very, very important. You have capability. Most of you have capability. But you make that capability realize its potential. That is by hard work. See, you have to have talent and you have to do hard work. Then you will have the beauty of science. You can witness the beauty of science. Nothing will satisfy you. No novel, no book can give you as much satisfaction as the beauty that you discover as a scientist. With that, I would like to conclude with thanks to CFTRI director and everybody else here for inviting me. This has been a wonderful experience. Thank you very much.
Thank you, sir. I think it was indeed a trip to Wonderland, starting from the importance of food proteins to complex structures that was elucidated at your lab. The multitude of proteins was so very well explained. Thank you so much. ಆಡು ಭಾಷೆಯಲ್ಲಿ ತುಂಬ ಉದಾಹರಣೆಗಳೊಂದಿಗೆ ಮತ್ತು ಸಿಂಪಲ್ ಆಗಿರೋ ಕಥೆಗಳೊಂದಿಗೆ ಮಕ್ಕಳಿಗೆ ಎಲ್ಲ ಪ್ರೋಟೀನ್ಸ್ ಬಗ್ಗೆ ಎಷ್ಟು ಅರ್ಥ ಆಗಕ್ಕೆ ಸಾಧ್ಯನೋ ಅಷ್ಟರ ಕೆಳಗಡೆ ಲೆವೆಲಲ್ಲಿ ನಿಮಗೆಲ್ಲ ಹೇಳ್ಕೊಟ್ಟಿದ್ದಕ್ಕೆ ಸರ್ಗೆ ಧನ್ಯವಾದಗಳು. ಐ ಆಮ್ ಶ್ಯೂರ್ ಫ್ರಮ್ ನೌ ಆನ್ವರ್ಡ್ಸ್ ದ ವೇ ಯು ಲುಕ್ ಎಟ್ ಪ್ರೋಟೀನ್ ವಿಲ್ ಬಿ ಡಿಫ್ರೆಂಟ್. ಪ್ಲೀಸ್ ಗಿವ್ ಹಿಮ್ ಅ ಬಿಗ್ ರೌಂಡ್ ಆಫ್ ಅಪ್ಲಾಸ್. ಥ್ಯಾಂಕ್ ಯು ಸರ್. Moving on to the next program. This year is being celebrated as International Year of Millets from the United Nations and the Food and Agricultural Organization, where India is playing a very indispensable role in this moment by taking initiative towards popularizing millets, whose consumption furthers nutrition, food security, and welfare of farmers. On this direction, CSIR-CFTRI had conducted an essay writing competition for the staff and students of the institute. May I now invite Dr. Padmavati Faustus to please take over and announce the winners.
With the aim to create awareness and increase production and consumption of millets, United Nations at the behest of Government of India declared 2023 the International Year of Millets. To commemorate this, Director CSIR-CFTRI has planned various events around millets. One such event was the essay writing competition on the topic India's Wealth, Millet Challenges and Opportunities for Using in Nutritious Diet, which was conducted on 21st February 2023. There were around 34 participants under 2 categories, scientific staff and students. Now, I would like to kindly request the chief guest and director to give away the prizes. Congratulations to all the winners and participants of this essay writing competition. The prize winners are the scientific staff. The first prize goes to Dr. Prakash M. Halami, Chief Scientist and Head, MFT and FPIC. Second prize goes to Mohammad Shakib, Senior Technical Officer, Grain Science and Technology. Now, next under the students category, first prize goes to Anjali P, Research Scholar, plant cell and biotechnology. Second prize goes to Raga Devi Sutari, Project Associate, Department of Biochemistry. Third prize, Harapriya Sahu, Research Scholar, Department of Biochemistry. And there are two consolation prizes. Mr. Calvin Gomes, dissertation students, fruit and vegetable technology. Second consolation prize, Zoya Sheikh, Research Scholar, Department of Molecular Nutrition. Thank you, sir. We would like to thank the director, CSIR-CFTRI, for the support and encouragement in planning this event. I would like to extend my thanks to the committee members for organizing the competition. Thank you.
Thank you, sir. May I now request a director to kindly give the presidential remarks.
Once again, good morning. And I think we'll all agree that it was really exhilarating to hear Professor Murthy talk about the wonderland of proteins. Having spent more than 3 decades in the field of proteins, I really enjoyed the talk very much. And although he did not know that the audience would be very young, he just brought it down to that level where they could understand, and it became interactive, which was a lot of fun to hear and enjoy. Indeed, however, his accomplishments are no mean feat, and really great science contributions were made by him. Now, coming to the National Science Day, as has been told, it was the discovery of the Raman effect, which is being celebrated since 1986 in India as the National Science Day. And I was just reading about Sir C.V. Raman and his paper after it was published. Apparently, 700 papers followed in 7 years. That was the kind of impact, although it was a very small effect as it was thought at that point in time, that light falls and the molecules scatter it and it becomes a different color. It was a very small phenomenon is what they thought, but the kind of applications that followed decades after, and today it is a kind of an indispensable spectroscopy that we use in every field. This is what science is all about. And one thing all scientists will agree with me, whether it is CFTR or anywhere in the world, is that we ask questions because of our curiosity. We follow that curiosity with a hypothesis, and then there is a high that we experience when we find out that we are right. If we are not proved right, then we go on into digging more and more to understand what is it that is happening. And that ends there to some extent. In institutes like CFTRI, we also try to find applications and go forward further to see how our newfound knowledge can be applied for the welfare, especially in the food area. So I was also thinking when I was sitting there, why did I even become a scientist? So even, of course, our teachers play a very great role in school. There are many youngsters over here. I'm sure many of you love science, maybe physics, maybe mathematics, because you have wonderful teachers, right? But science is also a very great equalizer. Like Professor Murthy showed, some of his students were girls, some of them were boys, but all of them did very significant work, and science does not discriminate. It's a great equalizer. So it doesn't matter if you're a girl or a boy. I think science teaches you that you can be on an equal plane. And apart from curiosity, of course, observation is very, very important. We observe very small things. Small children especially observe a lot of things. Sometimes the parents get irritated with that, teachers get irritated, why are you asking these silly questions? But nevertheless, it is, a scientist is probably a grown-up child who keeps the curiosity alive and goes on observing. And then they also try to follow it with a lot of tenacity and patience and document whatever those observations are. And finally, they communicate it. So this communication is also a great skill, which today we have been witness to the way Professor Murthy was able to communicate to all of us. So it is said that the person who convinces the world first gets the credit, not the person who found any phenomenon the first time. So scientists are also, it is very, very necessary that they become great communicators. And I think today our chief guest fulfilled all these criteria and gave us a great time. And we are indeed very, very thankful to him for having come all the way from Bangalore and adapted himself to explain a very, very complicated science that he had carried out in his lifetime to children. And I'm sure at least a few of you are very enthused to take science as a career forward. And just now when some of the youngsters were coming to pick up the prizes, he told them, let this not be the last one. Go on to bigger prizes. So probably that should be the take-home message. We look forward to see all of you achieve greatness. And yes, science is the greatest festival for us. Today is the greatest festival because it has no other barriers. And the last thing I would like to say is no matter where you perform your experiment, whether it is in India, whether it's in the US or in Timbuktu, in Australia, doesn't matter the geography, nothing. We get the same results. And that is why science is so great. And let us celebrate that today. Thank you.
Thank you, ma'am. Yes, science is the ultimate truth. Thank you, one and all. We have come to the end of this program. May I invite Dr. Srirama, Head PMC, to kindly propose the vote of thanks.
Thank you, Dr. Purnima. It is my privilege to have been asked to propose a vote of thanks on this occasion. On behalf of the directors, CSIR-CFTRI, and the entire organizing team of this program, I express our sincere thanks to Professor M.R.N. Murthy, Emeritus Professor, Indian Institute of Science, Begaluru, and also AstraZeneca Distinguished Professor, Begaluru, for accepting our invitation and delivering a highly motivated National Science Day talk. Sir, you have told a highly motivating, highly interesting story of your professional journey with proteins very beautifully and in simple terms to reach out the audience. As our director said, I am sure most of the children present here will get inspiration from your talk to become successful professionals. On this occasion, I request our director to felicitate Professor M.R.N. Murthy on this occasion. Chair. Yeah, thank you, madam. Thank you, sir, for accepting our felicitation. I wish to express our gratitude to Dr. Sridevi Annapurna Singh, our director, CSIR-CFTRI, for presiding over this program and providing all the guidance, support, and encouragement to conduct this program. I also thank H.S. Nagaraj sir. He taken out his time and participated in this program. I thank Dr. Nandini P. Shetty and Dr. Sri Arun Kumar for organizing the essay competition on millets, and Dr. Padmavati and Dr. Sri Somashekar for their help in help and cooperation in essay competition. I sincerely thank CFTRI Alumni Association for sponsoring the mementos for essay competition. I must mention our deep sense of appreciation for all the students and teachers for showing their keen interest to participate in this science program. I wish to express our sincere thanks to all the chief scientists and rest of the departments and the whole Jigyasa team for their support and help to organize this program. I also thank press and multimedia representatives for their support in covering this event. We thank our research scholars for singing a Vande Mataram during invocation, and my colleague Dr. Purnima Priyadarshini for nicely comparing this program. I thank the team of very highly motivated and dedicated members of the Institute event committee for organizing this event, and also staff of PMC, INP, and Computer Technology Services for their help and support in organizing this program. Thank you very much.
Thank you, sir. I request all of you to kindly stand up for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you. I request all the school students to please stay back in the auditorium.